All righty, we'll go ahead and get started. Welcome to Hats Off to the Human Brain. We are the Iowa Neurobank Corps, and today we'll be telling you a little bit about ourselves. So my name is Bailey.
I'm Peyton.
Hi, I'm Mara.
I'm Krina. Welcome.
First, I'll give you a little bit of an outline of what we're going to talk about. I'm going to present on what we generally do and what it means to be a research lab and kind of what research looks like. Peyton will be going through neuroanatomy and some brain origami. I saw someone said they really like origami, so hopefully you'll have fun with this. On that note, you can go ahead and get out your scissors and the sheet of paper with the colorful brain on it and start cutting that out and get your tape ready. Yep, Peyton's showing you if you can see it. It should look like that. Start cutting that out and getting ready to build it, because once we get to Peyton's part, you can start putting it together. And then at the end, Mara is going to have a fun little Kahoot for us to kind of go over what we've learned. And then hopefully we'll have some time to answer your questions at the end as well. We'll start a little bit about me. I'm a senior here at the University of Iowa. I'm studying human physiology, which basically just means I learn about all the different parts of the body and what they do. I'm on the pre-med track, so I want to become a doctor. And right now I have a research fellowship, which means the university has been sponsoring a project for me in the research lab. And when I graduate in May, for the next year, I'm going to be doing an internship with this research lab, which is really exciting because I get to stick around. And then hopefully I'll go to medical school afterwards. I got involved with the lab because a long time ago when I was a little kid, I've always loved science, and I've always known I wanted to do science. When people ask me what I wanted to be when I grow up, I would always just say a scientist. I didn't know what that would look like. But as I got a little older and realized I wanted to be a doctor, I started looking into what you need to do to become a doctor. And everything said, you need to get involved in research, do some kind of research. But as a pre-med student, you are pretty busy. And so I didn't want to get involved in something that I wasn't going to love and be passionate about. So when I found the listing for the Neurobank and applied, I was just in love. Like she said earlier, you want to find your spark for science. The Neurobank really does that for me. It helps me remember why I love science and why I enjoy science. And on the bad days, I remember that's why I'm here. Just to warn you, there will be pictures of the human brain ahead. So if that's scary or grosses you out, you can close your eyes for a little bit. There's only a couple slides with those pictures on it, but I just wanted to give you a warning. So like I said, here's our human brain. One of the things we do is we take care of human brains. So when a patient passes away at the University of Iowa, they can choose to donate their brain to science. And what that means is we take the brain and take care of it and process it. And by that I mean we cut it into different pieces so we can research specific pieces of the brain. Something we call ourselves at the lab, we call our lab kind of like a library, and we're the librarians, which means, you know, when you go to a library, all the books are organized. You know, if you want to find science or history or fiction, you have those regions you can go to. We kind of do that with the brain. If we want this region, we look here. If we want this region, we look here, or a specific disease, things like that. So we make sure to keep our brain bank organized so that we can find things. So next to me, you can see Mara. She's helping get things organized. She's using tubes to put specific parts of the brain into them. So when we need to, we can find them. In the back of the picture, you can kind of see Sadie. She's working in something called a biosafety cabinet, which is a special cabinet that helps you do experiments. So what she's doing in there, when a brain is processed, you can take special parts of it and grow cells from it, which is really exciting when you want to study diseases, or you can kind of do medicines or therapies. You can learn all sorts of things from these cells, and it's a really exciting advance in research. So a little bit of a closer look at what brain processing looks like. I'm cutting a brain here in the big circle. I use a really long blade and cut really thin slices of the brain because you have a ton of different.
Long blade. It's like
it is a very long blade
for a brain.
It's a special blade.
No.
So we want to be able to see all the different structures you have. And to
I
do
have two
that,
of them.
you can cut thin
Second one
slices
for a different trigger.
so you can
and,
see
Jerv-
all the different parts of the brain. And once you cut those slices, we can organize them onto the slab you can see there. So then when we want to go looking for those structures, we can find them. And we use a really nice camera to take pictures so we can catalog everything we have, and we can kind of have like a database of where all our important parts of our brain are. So the brain we were just looking at was a fresh brain, and we freeze it. Another type of research we do is a fixed brain. So what that means is we use a special liquid called formalin, and that helps to preserve the tissues. You can have tissues for years and years and years kept in formalin to continue to research them, which is really wonderful when people want to donate their brain. Their body can help people for years to come. And with formalin, we have those same slabs that we had earlier, and we put them back together kind of like a puzzle to find all the different regions that we need to study. In our name, we're the Neurobank Core, which means we're a really big team. We have lots of moving parts, different projects all running at the same time. So every week we like to come together and share things. We share the good things that have been happening, the not so good things that have been happening, new exciting projects. And another exciting thing we do is called Journal Club. Journal Club is kind of like reports you do for school, but more fun because you get to pick a topic you're really interested in and share it with the group. So last semester, I picked a really almost old research article on Alzheimer's that had kind of a historical perspective, and Mara picked a really new, exciting research that's happening with Alzheimer's. And so we got to see a very long spread of the disease and kind of put all these pieces together and learn as a group, which was really fun. With my fellowship that I talked about earlier, I got to take my project to a fair and present to all sorts of different people. So this was a university-wide fair. So there were history majors and art majors and math majors, and we all had our research that we're all working on and had our posters, and we were standing up there. There were hundreds of us, and we would all just go around and learn about everybody's different research and what's going on in the university. It was a really exciting experience to both share my research and learn about other people's, and work on scientific communication also. My project is on COVID and the brain, and we're looking to see if there is an interaction and what the interaction there is between the disease and our brain. So we've kind of seen a big picture of our research lab, and now I'm going to show you a few things specifically that we can look at in the brain and what it means to actually research the brain. So we can use a microscope to, okay, so we had those slabs of the brain. Well, we can get even smaller, like 5 micron is the size. And it's basically smaller than your fingernail. If you look at your fingernail, it's even smaller than that. And it's a slab that we can put on a slide and look at under a microscope. And you guys probably know your whole body is made of cells. Well, sometimes those cells go bad, and that can lead to disease. So when we have these tissues on a slide in a microscope, we can use something called a stain that makes sure we can see the bad cells. So then here you can see these red speckles. So those are the bad cells in this brain, and they're causing a disease called Alzheimer's. So we can see these things in the brain and see what's causing the disease, which is really exciting. This practice has been done for a long time to see what's happening in brains. A new exciting practice is that we can see multiple bad cells all at once. So in the last, we could see the red splotches. Well, now we can see red and green and blue. All these different colors are different types of bad cells that are all working together to cause a disease. So then we can learn more about the disease than just one cell. We can learn all sorts of things about the disease. Or if it's a healthy person, we can see what cells are supposed to look like, which is really good too. So now I'll pass it on to Peyton.
So hi, you guys. I hope by now you have this mostly cut out. If not, that's okay. I do recommend, though, that you guys turn on your cameras. That way, if you need help, we can actually see where you're at and help you fix the brain. But a little bit about me before I start talking about the anatomy of the brain is that I am also here at the University of Iowa, and I'm a sophomore. I am a neurobiology major, and I am also on the pre-med track, which basically means that I just do a lot of biology and I want to become a doctor one day. So I'm actually from around here. I was born in Monticello, Iowa, which is about an hour away from Iowa City. And I knew since I was little, I really liked science, and I always really enjoyed every science class that I was in. I even remember growing up and like that being the only class I was excited for in school. And so as I got older, I kind of realized specifically how much I liked biology. And then when I got to college, I was able to study it more. And I've kind of just fell in and found that I really like learning about the brain as well, which is what got me here. So moving along, a couple of images as well, like before. This is that cell growth that we were talking about before. And basically what we're doing here is in the little circles in there, it's kind of hard to see, but we're using something that Dr. Lynn likes to call cranberry juice. And it's not cranberry juice, I promise you guys. It's a it's a hardcore chemicals that we put into these that help them grow into new cells. And we're actually using dead tissue to grow new cells, which is crazy that you can take something dead and make it alive again. But that's what we're doing here is we're actually looking at those cells and seeing how much they've grown since the last time we've looked at them, which is really interesting and something that not a lot of people are able to do, let alone see. Which moves us on to something that I've been able to personally do is use this technique and make it into sort of a business plan and in hopes that it would be for more personalized medicine. So one person can see their own cells and how they react with specific things. And so I was able to enter into a competition at the business school here at the University of Iowa. And I'm not in the business, like no way, but I was able to win some money for the for our lab using the techniques that we've already established and just building further on it. So you can really do anything with science. You can go into any realm doing really anything you're specifically passionate about. So the neuroanatomy, this is the fun part in my opinion. And I'll start with giving you just a little brief kind of introduction. As you can see, there's two brains here. It's the rat and the human brain. And if you think about these two types of brains and think about the two types of animals, rats are significantly less advanced than we are. Like we can have conversations with each other and you know, have complex thoughts and do all these different things that a rat can't do. And part of that's attributed to, and the majority of it is the fact that our brains are much, much larger. Having a larger brain allows us to have many, many more of these cells that allow us to create those connections within our brains to understand what's going on. So our brains, human brains, are actually made of 100 billion nerve cells, which is crazy if you think. I can't even honestly conceptualize how many cells that is. It's just a crazy amount. But what makes it so, the ability for it to have so many is those ridges on the brain called sulci. And these sulci allow for more surface area and allow the brain to have more room for more cells. And you can see the rat brain doesn't have those divots. And that's why they don't have as many cells as we do. So I'm going to talk about parts of the brain as you guys are going to fold. So we're going to move to the next slide and have the directions. But just keep in mind while you're doing it, I'm going to explain some things, okay? So to start us off, I'm going to start in the frontal lobe, which is right here in your brain, and it's the pink part on your little cutout here, okay? So the frontal lobe is in charge of things like voluntary movement, expressive language, and something called executive functioning, which is basically like higher level actions. So like an example of this would be being able to organize things. So how many of us, and be honest, how many of us have clean rooms right now? Okay. Well, mine is messy. I will be honest. Mine is very messy. And that would be my frontal lobe in charge of that function. My messy room is because my executive function isn't that great. So yeah. Next up is the parietal. Yeah, what's up?
I actually organize my collectible card collection all the time because
you know
it's
what?
just not right.
Your executive function is working very, very well. And that means your frontal lobe is doing an excellent job. And I wish I had part of yours. So Moving to the back of the brain is semi in the back is the parietal lobe, and that's right about here. It's the yellow part on our little diagram here. And the parietal lobe is in charge of your touch and your pain and your body's temperature. And so how many of us have broken bones? How many of us have sprained our ankles, broken bones, doing anything like that, gotten hurt? Yeah, me too. I've broken my collarbone 3 times. So I know that pain. I know that feeling. And so those pain and those like, ouch, I need to go to the doctor. Like I know I'm in pain right now. That is in your parietal lobe. The next part of your brain is more in the back, and it's the tan, and it's kind of orangey. It's the occipital lobe. And this is in charge of just vision for the most part. Your temporal lobes are the green regions over here and here and here. And these are centered right about here in your brain. And as you can imagine, being centered right around your ear, it's in charge of a lot of your hearing and processing with the hearing. It's also in charge of a lot of your emotions and your memory and even language. So, you know, there's a lot of important aspects along with that. Moving on to the cerebellum, which is our blue part, and it's resting a little bit lower on our heads. That is in charge of your coordination and balance. How many of us have like spun around in a circle really, really fast and then gotten super, super dizzy and like almost fallen over, like playing games and stuff? No. Well, I have, and I know I have gotten really, really dizzy, and that would be your cerebellum in charge of that dizziness and your brain being, and it's that part of your brain that's keeping you balanced and aware of how you are in the world. So moving kind of onto the bottom part, which you guys are making here, is there's a bunch of different structures on the bottom so that are really, really important for very specific parts of what your brain does. So to start, we've got that olfactory bulb, which is in charge of moving the signals from your nose all the way up into your brain. And those sensations of your smell. So that moves your smell up to your brain. Your optic chiasma does the same thing, but with vision. It moves the signals from your eyes up into your brain to process. The pituitary gland is my favorite, and it is about the size of a pea in real life in your brain. And it's kind of like a mailman. What it does is it sends out all these hormones all the way to the rest of your body, and like packages. And the recipients of those packages, which would be things like your skin or your mood or your eyes, then are using these instruction manuals from the hormones to just do what it needs to do. It's really interesting how it all works and how it all interconnects in your brain, and all just a little bitty thing is what does all that. Moving kind of down backwards in the brain is you've got your pons and your medulla oblongata, which is basically like a highway between your brain and your spinal cord. It'll make sure that the signals and all other kind of sensations are moving from your brain all the way through your spinal cord and out through your limbs, and everything that happens in your limbs goes up through your spinal cord and up and back into your brain. So it's really important, like connecting everything. So I hope that you guys are done and you've completed it pretty well. Are we seeing, is it pretty good? Let's see. I'll take a quick look. Somewhat. Okay. Nice job, you guys.
All right. Those brains look good. My name's Mara. I'm also an undergrad here at the University of Iowa. I'm a neurobiology major like Peyton, and I have a chemistry minor. I'm also on the pre-med track, so I hope to go to med school, and I want to work with kids and the brain. So how did I get here in research? I'm originally from Des Moines, Iowa, and I have always been really interested in science too. I liked reading a lot of books with animals. I liked marine life and things like that. And when I got older and in high school, I took a psychology course, and I just really fell in love with the brain, and it really interested me. So when I got to college, I knew neurobiology, and this lab sounded right up my alley. And so that's kind of how I got here. So, all right, we're going to do a Kahoot. How many of you guys have done a Kahoot before? Does anyone know? Yeah. Okay. It looks like everyone's done it. Okay, perfect. I'm gonna go. All right, I'm gonna go ahead and start it. If you're not in, you can still join. The game pin is in the chat, so you can join at any time. All right, so the first question, how much of your consumed energy goes to your brain? So this is just a fun fact. You're not expected to know it, just take a guess. All right, good job, you guys. Wow, that was really good. About 25% goes to your brain, which is kind of crazy because your brain is definitely not 25% of your body, but it takes a lot of energy for all your thinking. All right. Question number 2, which of these is a cell in the brain? Now this is kind of a tricky one. So just make your best guess. Don't have to be right.
Not yellow.
Yeah, I mean, could be. Okay, yeah, nobody chose cell phone, so that's pretty good. But some of you guys got it. Yep, so renal cell, that's in your kidney, actually. An islet cell is in your liver and pancreas, but the astrocytes, those are actually cells in your brain, which is kind of hard because it didn't say cell, but site does mean cell. So that's just another little fun fact. All right, how much does the average brain weigh? So this is an adult human. How much does an average adult human brain weigh?
I know this one.
Oh, you know it? Well, then don't tell the answer yet. All right. 3 pounds. How did you know this one? Have you heard it before? Maybe? Yeah?
Yes,
Yeah.
but maybe it changed.
Could have. Well, it's about 3 pounds and it's about the size of your 2 fists put together. So it's maybe a little bit bigger, but that's a guess. All right, Carl in the lead. All right, what is the oldest part of the brain? So which part of the brain developed first? And this is kind of evolutionarily. So which part?
I think I know it.
Okay. Got everyone's answers in. All right, yeah, some of you got it. So the brainstem controls a lot of really important functions like heartbeat and breathing and things like that. So that's got to come first. So you can go ahead and do those things to continue to evolve. All right. So now this is some of Peyton's talk. What is the brown region of the brain? So right back here. What is that, or what is that called?
Please don't answer green.
And it's not actually brown in your real brain, which is, it would have been cool if our brains are color coordinated, but. Good job. Almost all of you got it. Yes, your occipital lobe, important for your vision. All right, Carl, you have a streak. You're doing really good. All right, what does the green region interpret? So what is the green region, your temporal lobe right here? What does that do? Right. Hearing and speech. Good job, you guys. You guys were really listening to Peyton. That is really good. All right. What are the folds of the brains called? What are the little divots, remember, that humans have that rats and other animals don't have?
It has been mentioned today.
Right. The sulci, yes, very good. Fissures are something in the brain, but not exactly the sulci. All right. Now the rest of these are going to be true or false. So the right side of the brain controls the left side of your body. Is that true or is it false? What do we think? What do we think? All right, it is true. Your brain is called contralateral. So that means that it controls the opposite side of your body. Okay. Your brain is not active when you sleep. Is that true or is that false? Ooh, that one was quick. You guys all knew this one. Yes, that's false. Of course, your brain is active when you sleep. You use your brain for lots of different stuff, including dreaming, which is really important when you sleep. All right, last one. You use only about 10% of your brain. True or false? Yes, good. False. You use 100% of your brain. You need your brain for all sorts of stuff and you can't get rid of any of it. It's all important. All right, let's see the podium. Got Cara in third. Good job. I am cool is in 2. And Carl, good job. I mean, everyone did a good job. You guys did really well with this one.
I was fourth. Yay!
Good job. All right. Now we've got a few minutes left. If you guys have any questions, now's the time to ask. Anyone have any questions?
Even comments, anything.
Mm-hmm. Yeah.
Do you just study human brains?
No, so in neurobiology, that's a great question. In neurobiology and things like that, you can look at a lot of different brains. You need to look at all types of different nervous systems because sometimes you have to look at other animals too to learn about the human brain. So
like a cat?
I haven't looked at a cat's brain, and I don't think we use cats all the time in Neurobank. But we do use mice brain a lot in the Neurobank. We look at
And pig. Pig is actually very similar to humans.
Yeah, pig is similar to humans. I know some other neurobiologists use fruit flies. They actually look at the fruit flies and their brains and can do MRIs on them and some cool stuff like that. So yep, lots of animal brains.
How does dementia work?
Good question.
That is a loaded question.
Yeah. Yeah, Bailey, you take this.
I'll give you the short, basic answer, but basically, dementia is a symptom. So there are all sorts of diseases that can actually cause dementia, and dementia just means memory loss or loss of cognitive functioning, which basically means your brain isn't working right. So there can be lots of things that cause it, but most of the time it's because things are causing the cells, like how I said bad cells earlier. The bad cells are becoming too much in the brain, so there's too many bad cells, or something else is attacking the cells and making cells die. So you know how we talked about the brain being 3 pounds? In people with dementia, their brain usually weighs a lot less because they've had a lot of cells die. Did that help? Okay, I see there's another question. Go ahead.
So what is Alzheimer's in a quick 2-minute answer?
I can keep going if you want. So Alzheimer's is a disease that causes dementia. So like he asked how dementia works, one of the symptoms of Alzheimer's is dementia. So that microscope slide that we looked at, that was actually a slide of Alzheimer's. So those bad cells had accumulated, which just means there's a lot of them, and they were causing Alzheimer's, which when the brain isn't working right, that's kind of what causes the memory loss, like I was explaining earlier. Was that a simple enough answer? Did that help?
think what's also really cool though about Alzheimer's is there's a lot still that we don't know. And that's a lot of why we study the brain, is because there's so much that we just don't know yet. And so much that like is interesting to learn. So yeah, there's a whole bunch of different, parts to it, being the dementia and, there's, the cells dying, but yeah.
All right. I think we're going to get sent back to the main room, but thanks for listening, everyone. It was nice seeing everyone today.